Our first talk in this session is given by Zheng, and she is from the laboratory of Professor Mala Pudine from Electrical and Computer Engineering Department, and she will be talking about hydrogel microneedle assisted assay integrating aptamer probes and fluorescence detection for reagent-less biomarker quantification. Okay, then, please come over.
Hi, everybody. I'm Hanjia Zheng. I'm from ECE Department of University of Waterloo, and my supervisor is Dr. Mala Pudinates. So today I'm going to talk about my master project. So first of all, I would like to start with the definition of point of care biosensors. So for the recent decades, the point of care biosensors has been widely applied in our lives, such as glucometers and pregnancy test strips. It is a type of easy to use and rapid medical diagnostic manner performed at or near the patients, which allows for patient self-administrations. The common sample source for point of care, such as blood and urines, are relatively hard to obtain. Others like sweat and salivas are easily getting influenced by the environment. Recently, there's a new type of biomarker sample source for point of care called skin interstitial fluid, shortened as ISF, catching its rising attentions recently. So skin ISF is a major component of body fluid. It is located in the epidermis and dermis layer of skin. So as we can see in this image, the ISF surrounds the cells and capillaries. It acts as a reservoir holding the nutrients diffused from the capillaries and the waste product generated by the cells. Therefore, it contains wide range of biomarkers. So why ISF is considered as a great sample source for point of care? So first of all, it contains a wide range of biomarkers, including proteins and nucleic acid, or also some metabolites such as ATP and glucose, also some drugs like heroin and antibiotics. According to this Venn diagrams on the right side, the biomarkers existed in the blood can also be found in ISF. There are 60% of them. And also 29% of biomarkers can only be found in ISF, which is a huge amount of them. Also, ISF can be a good sample source for long-term continuous sampling manner because it doesn't contain clogging factors different from blood. So as my introductions, there's many advantages of ISF compared with other type of biomarker sample source, but why as ISF hasn't been widely applied in clinical usage? So the major obstacles is the ISF sampling manners. The conventional ISF sampling manners targets the ISF deeply located in the skins. So the sampling process is time consuming and it will cause tissue damage. However, here we found a new type of ISF sampling manner called microneedles. It targets the ISF in the epidermis and dermis layer. So microneedle is an array of mini authorized needles down to micrometer scales. It can penetrate through the skin and access to the ISF located on the epidermis and upper layer of dermis. So it can collect the ISF without touching the nerve ends and blood vessels deeply located in the dermis layer. So it is considered as a pain-free and minimally invasive manners. So commonly, microneedles can be divided into three types: hollows, porous, and hydrogen microneedles. Compared with hollow and porous microneedles, hydrogen microneedle is simple and quick to fabricate because it contains one or multiple type of polymers. And also it has great biocompatibilities. In addition, it can collect higher volume of ISF in a short time point. Here, I would like to demonstrate a hydrogen microneedle-based reagent-less fluorescent assay for minimally invasive detections, shortened as RFMIDs. These RFMID devices are fabricated by the methacholated hyaluronic acid, and we utilize aptamer switch probes with a strand displacement strategy for target detections. The aptamer probes we're using at here is a short single-stranded DNA probe, and it can bind to some specific targets with great affinity and high specificities. The target of interest we detected here are some metabolites, including glucose and ATP, and a common mimic of L-tyrosine called L-tyrosinamide. And also we detect the type of protein called thrombin here. It's also an important biomarker for hemostasis and blood coagulations. Next, I would like to briefly introduce the polymer we're using it here. It's called methacrylated hyaluronic acid. It's a chemically modified hyaluronic acid derivative. Compared with simple HA, MEHA has a carbon-carbon double bond, which can be covalently linked to the other MEHA molecule under the UV treatment. It has great biocompatibilities, and the cross-link network under the after UV treatment can provide great swelling ability of the microneedles. Therefore, it can collect the ISF effectively. Utilizing the same cross-link reaction, we can immobilize our aptamer probes on the MEHA matrix. So in this project, we modify our aptamer probe with the acridide molecules, which also contains a carbon-carbon double bond. So it can covalently attach on the MEHA matrix and form a very rigid bonding. At the bottom is a chemical overview of our microneedle patches. So you can see the linkage between the aptamer probes and MEHA matrix and the cross-linked MEHA network. Next, let's move to our sensing strategies. So our sensing system, including two components. One is first is the aptamer probes labeled with the fluorophore, and its complementary strand labeled with a quencher molecule. So when the aptamer probes hybridize with its complementary strand, the fluorophore is bring close to the quencher molecules, so the system's fluorescent intensity is low. After the microneedle capture the targets, the aptamer probe binds to the targets and denatured with its complementary strands. So at this moment, the system's fluorescent intensity is high. So in the actual experiment, we immobilize the aptamer probe and its complementary strand complex on the microneedle matrix. The measure is fluorescent intensity before and after target capturings to calculate the target concentrations. So usually higher fluorescent intensity difference before and after target capturing indicates greater target concentrations. So the picture on the right side is the microfluorescent microscopic image of our microneedles. So we can see the brightness of the image is proportion to the fluorescent intensity of the microneedles, and we can see the fluorescent intensity of the patches increasing with the rising target concentrations. Next, let's move to the RFMID fabrication process. First, we prepare a MEHA solution contains cross-linker and photoinitiators. Then we add aptamer and its complementary strand complex on top of it. After the patch was completely dry, we do UV treatment and wash the patches with buffer solution to remove the unbounded aptamer strand. Eventually, we will get a microneedle arrays with a needle length of 800 micrometers. And here is an SEM images of our patches. We first test our devices on the agar and porcine skin contain certain concentrations of targets. Then we move to the animal experiment. So here we built up a diabetic rat animal models for the experiment, and we apply the RFMID patches on its back skin. So to note that the whole patch applying process, the rat was not under anesthesia, which indicates our microneedle is a pain-free manner. And after we remove the RFMID patches from the skin, we can see there's an area of micropore generated by the microneedle penetrations, which indicates the microneedle can penetrate to the skin effectively. And this micropore can be held in 15 minutes after the patch was removed from the skin, which further indicates the microneedle manner is minimally invasive. Next is the glucose capturing efficiency experiment. So here we use the diabetic rat models, and we measure its glucose level, initial glucose level with glucometer and the RFMID patches. Since it's diabetic, so its blood glucose level is really high, greater than 30 millimolars. after that, we inject insulin into the rat, then its blood glucose start to decrease. During this time, we measure its blood glucose, track its blood glucose level with glucometer and RFMID patches until the rat reached to hyperglycemia range. Next, we inject the glucose solution into the rat, and its blood glucose will start to increase until it reached to the normal region. So the graph at the bottom shows the results we get from both RFMID patches and glucometers. So same as our expectations, the initial glucose level is really high. After insulin injection, it decreased very fast and hit the bottom. After it hit the bottoms, we inject the glucose and the glucose level is rise back to the normal. So we can see the results we get from RFMID and glucose is close but slightly different. This is because the glucose level in the blood, in the plasma and ISF are slightly different during the glucose level changings. So the appearance of this, the appearance of the lag time between blood plasma and ISF can further indicate the reliability of RFMID patches. At the end, we compare our RFMID devices with other type of microneedle biosensors. So as we can see in this table, all of the other microneedle biosensors requires a long target capturing times and a very complex post-processing steps, including washing, blocking, and detections. However, our RFMID patches only requires 2 minutes for both target capturing and detection, which is significantly lower than the other type of manners. So overall, I think this RFMID devices can open some new avenues for microneedle biosensor development, and it has great potential for manufacturing and commercializations. Here I demonstrated 4 competitive advantages of RFMID devices. First, the fabrication process is very simple and quick, and also it only requires 2 minutes for target detections and ISF samplings without any post-processing steps required. At the end, it could give us very high accuracies on the detection results. So overall, I think this RFMID patches is a valuable research directions for us to go. So in the future, we are trying to solve some problems for RFMID patches. These obstacles are mainly the obstacles for all the fluorescent-based biosensors, such as fluorescent microscope is required for signal detections. Also, it cannot be able to do continuous measurement because the fluorescent signal requires a longer time to switch the structures. And things we want to improve in the future is first, we can develop a cost-effective and mini-authorized fluorescent detectors for which we can use our smartphone camera to detect the fluorescent signal. Or we can switch our research directions to electrochemical-based hydrogen microneedle biosensors, which enables for continuous measurement. And appreciate for everyone works in this project. And here are my references. Thank you guys for listening.
Thank you so much. It was very on time. Perfectly done. We have time for 2 or 3 questions from the audience, and please, our online audience, remember that you're always welcome to ask questions. We can read them aloud here and ask any questions out there. I had a question for you. The microneedles that you develop, how robust they are? Would they last multiple tests? In general itself, of course, fluorescence probably just one-time test, but in general, how robust they mechanically?
Fortunately, currently, this is one-time patches measurement, but we did some stability tests for the RFMID. It can last stable for 14 days storage under room temperature.
Okay.
Yes, but currently we cannot develop a very robust patches for multiple testing. I think that's one of our future direction as well. And I believe for other type of microneedle based biosensors, like for electrochemical sensing, it can last for multiple tests because we want to do continuous measurement.
Okay, then. My second question before you pick, you were specifically showing the data for the glucose monitoring.
Yeah.
What do you think could be next couple of biomarkers that very valuable to add on at time?
Sorry.
In one of the slides, you showed multiple biomarkers that could be explored through ISF approach.
Yeah.
You showed results for glucose. That's a good starting point. What else would you add if you had a chance?
Oh, we already test like all the 4 biomarkers I listed on the slides, like glucose, ATP, and L-tyrosinemide and thrombin. So all of them has got very good performance and very accurate readouts from our RFID patches. But I believe there are going to be more molecules that can be detected by this kind of approaches. So for instance, my lab mates are trying to detect serotonins, and vancomycin was the same, like relatively similar approach, but also the target of interest or target that can be detected by this approach are limited by the aptamers, because it have to be like aptamers, great aptamers available for it. Yeah.
Okay, thank you. Any more questions? Okay, at that we will continue through our agenda. Thank you so much. Our next speaker today is Lukas Bauman from Laboratory of Professor Hanjia in the Chemical Engineering Department. A little shorter title, so it's easier to read. 3D printing personalized medical hydrogel masks. And I think, Lukas, you all set? Go ahead.
Multifunctional hydrogel masks and wound dressings towards digital health and personalized medicine. So the overall objective for this was to create a, that doesn't seem to work. The overall objective for this was to create a double network hydrogel that has multifunctional behavior and isopropyl acrylamide to create a mask that can really when applied to a patient, so that you can create complex geometries for fitting like noses, fingers. A typical wound dressing would have difficulty fitting on that, as it's a 2D plane. So what is polyanisopropyl acrylamide? So this is a homogeneous polymer that has a critical solution temperature. This polymer switches from hydrophilic to hydrophobic above 32 degrees centigrade, and it's very common in microgels delivery and also for things like cell culturing, where you can grab a brush structure and then create a full sheet of cells once you cultivate it and reduce it below 32 degrees. It also has a tunable through copolymerization, where increasing the hydrophobic portion decreases the and adding hydrophilic increases that temperature. So what is the motivation for this 3D printing? Well, tips of making hydrogels limited by the casting geometries. You have to make a mold for every size that you want to cast. And into this, you're limited to 2.5D structures. You want the width to get lower as it goes down the mold. You can't have full 3-dimensional structures with traditional casting. Or very printing is because make whatever structure you want if you have ideal support. But in the past, it's been limited by the machine cost and the resolution, where bio-ink 3D printers typically can't go below 100 micrometers because they are limited by the size of the needle that you print with. In addition to this, the so recently we have seen significant improvements with 3D printers, such as down significantly. So we wanted to do was create this hydrogel with a resin 3D printer, and we wanted to use sodium alginate to reinforce the hydrogel. And that's because polynipam is typically very weak if you just cross-link it, and once it's fully swollen. But the issue with sodium alginate is that in resin 3D printers, are viscosity minimized, sodium viscous, and you can't typically print it in water. And in addition to that, it's not really soluble in other than water. So we'll just look quickly at other examples. So here we can see other examples of polyanisopropyl acrylamide hydrogels, and we can see that here they are either solution but very small, resolution and in addition, second, they don't really show any drug release because it's so heavily cross-linked. You have to also reduce the amount of cross. Still see that change in the gel. So how we did this was we made base, but we made this in a oxide that is aprotic. And how we did this was we dissolved sodium alginate in water in the presence of glycerol dimethyl sulfoxide. The glycerol allows the sodium alginate to remain stabilized in dimethyl sulfoxide once water is evaporated through rotary evaporation. So the network, once we had this base, we add a cross-linker, which is pluronic diacylate. Pluronic is a polyoximer, and this is a tri-block copolymer of polyethylene glycol, polypropylene oxide, and ethylene glycol. This has a separate lower critical solution, so we can actually imbue multiple thermal responses, different temperatures within the material. So it has separate states of shrinking. In addition to this, we have the poly or N-isopropyl acrylamide, which is shown in dark red, and then the alginate once it's cross-linked network in yellow. In addition to this, to facilitate and help the dispersion of the sodium alginate in the resin base, we have acid coated, which are due from tannic acid and CNCs in the slightly basic solution. Enables the resin to be very homogeneous and print well while the CNCs add to the reducing warpage printing. So now we look at the personalized mask. So in general, with these masks, with wound dressings, they don't have any medical value themselves, but what they are good for is a matrix for personalized medicine. So you have this through its, you can load it with any drug you want, hydrophilic, or if you have a nanoemulsion, which we haven't tried, you synthetically with hydrophilic drug somewhat. So what we do is we use 3D phone called Scandy. We scan someone's face, and then we edit it. So we remove the eyes, nose, and nostril and mouth, and extrude it out to generate this personalized 3D mask. And then we add structures to the printing of it through supports. Here are some of the examples of our 3D printed objects. And so these are very small. They're actually 10 millimeters, but the feature size, see here from the top, the precursor is very high resolution, and that resolution over calcium chloride. In addition to this, we can see that the structures seen in the precursor and the final 3D model, the back model, and the that are angled with resin 3D printing are incapable of printing these features. But note here is that the printed part we around these issues. So next we'll look at the thermal response of swelling of this material, and here we can see a where 9 degrees and 32 degrees a midpoint volume, and after that it's at a low volume. And here we can also see on the left that on the left the material is very repeatable, with it able to swell and deswell over multiple cycles. This indicates that this is reusable, so it could be reloaded, sterilized, and used again for the same patient, enabling the recyclability of the material. In addition to this, so the important thing for this material is that the reason we chose to have 2 lower critical solution temperatures is that when it's used at room temperature, it's going to be stronger than the at 4 degrees. And this is very important because polyanisopropyl acrylamide, when it's fully swollen, is an extremely brittle, weak material. And you don't want it to break while you're applying it in the field or to a patient. So for this reason, we added a secondary thermoresponsive crosslinker. We can also see here that from the SEMs, it goes from porous to very non-porous, where it's very porous and almost a spiderweb structure, and then it becomes more globular as the temperature increases with the hydrogel shrinking and becoming a cohesive structure. In addition to this, we can see that the hydrogel is quite strong in compression, and the precursor hydrogel is very strong at 14 MPa maximum tensile strength. This is a benefit because it means you could actually print this material off-site, ship it to the point of care, and you could have a fabrication facility that enables you to print this without having a hospital or a point of care have to set up these systems there. Next, we show the smart adhesion of this material. With a simple treatment, we can increase the adhesivity of it at 40 degrees or body temperature. And in compression and tension, the material shows itself sticking to the hand. And then next, we have just a video demonstration. And then here we can see it does stick to the hand and it has to be peeled off to remove. It doesn't easily fall off, which is ideal for a hydrogel wound dressing. Finally, we have a very qualitative assessment of the drug release. We do have some more data for this, but it hasn't been fully collected. But here we can see there's initial burst release when the cube soaked in rhodamine is dropped into 37-degree water. And then over time, the color density of the solution increases, indicating a release over time. So with all this, the idea for this material is to create a matrix that could be loaded with different drugs for personalized healthcare and for patient-specific wound dressings, where depending on the surgery or the person coming in, you could develop this to give to a patient and create something that won't be as generalized as typical wound dressings. And we also design something that could deliver drugs that you want to deliver. Thank you for listening to my talk. Again, I'm under Dr. Boshin Zhao, and our research group is the Surface Science and Bio-Nanomaterials Lab. I'm open to any questions.
Thank you so much. It was again very well done, all on time. We've got extra time now. Thanks for your performance here. We could have 2-3 questions. And Emmanuel, please.
Thank you, Lucas, for a wonderful talk. I just have 2 questions. First question, the thermal responsive nature of your product, I guess. Have you considered regions around the world where ambient temperature is a lot higher? How will this product be able to be utilized for patients in those regions?
So for this, well, you can use that with, as long as it's not above body temperature, it would be okay. But obviously this would not be suitable if the ambient temperature is above body temperature. You can modify it. So hypothetically, we could add something like hydroxyethyl methacrylate, which is a very hydrophilic acrylate monomer, and that will increase the LCST. So it would release closer to body temperature versus at 32 degrees.
Thank you. And the second question, the adhesion of your wound dressing. Do you think that will be a concern for some patients, because the adhesion seems to be quite strong? I mean, that is one of the challenges with a lot of wound dressings, is where after you apply it, and then when it's time to remove, you may actually, I guess, cause further damage as well. Do you think, have you actually considered that or looked at it? I'm just wondering what your thoughts are.
So we are developing further adhesive strategies. Right now, it was generally designed on degrading the top layer to make it adhesive. And this does show some smart adhesion, but the adhesion is actually not that high. It looks very high for a thin film, but when it's as a 3D printed object, it's not as compliant. So it does stick, but it is not as adhesive as the demonstration shows, as the material has higher cohesive strength. So it doesn't conform to the surface as well, which is positive and negative, depending on how you look at it.
I love your consideration for reloading and reuse. I see this as a really great solution for patients post-op, or anyone that's taking care of someone that's just had a surgery. You know, they go through so much gauze, so much tape. This could be a really great solution. What would be the strategy for reloading a drug for someone?
So it might be more difficult if you're changing the drug, but in general, just reloading would be after it's used on the patient, you would sterilize it with something like ethanol. You could also do an ethylene oxide sterilization. We haven't tested that, but it could be a possibility, though definitely not autoclaving. But after that, you just put it in the fridge in a solution loaded with your drug. And if it's hydrophilic, it'll pull it back in. And that's the beauty of this, because it can be shrunk and swollen multiple times. You can reuse this indefinitely. From our tests, we had, I think, 12 or 15 cycles of changing the swelling of the matrix, and it still shows very reasonable results. Though this could also be modified. One of the issues with this matrix is it is a calcium cross-linked alginate, and over time, the alginate will lose its calcium ions. So by re-soaking it in calcium chloride to recross-link the matrix, you could regain the original behavior of the matrix again as it loses that behavior.
Thank you so much.